policy that the affirmative side is advocating wouldn't work because it limits the progress of all other stem cell research. Jonathan Moreno from 8 Reasons to Applaud Action on Stem Cells says the ultimate goal is to develop a guidebook that will tell us exactly how each gene or combination of genes contributes to the development of a unique individual. This will greatly enhance our understanding of basic genetics and could allow scientists to develop drugs that can prevent the diseases from developing in the first place. But they can only do this by tracing the development of human cells from the very beginning. Therefore, scientists need to conduct research on embryonic stem cells so that they can discover how these all-purpose cells can change into any one of the more than 200 cell types in the human body. So you need to do more embryonic stem cell research to be able to understand or to do any other type of research on other stem cells, like the one that they're doing on the stem cells, like adult stem cells that their policy was using. And Michael Runeta of the Life Science Crucible says, additionally, embryonic stem cells can aid in the refinement of aging drugs since the cells can be differentiated into specific cell types on which scientists can quickly test whether a drug has a desired effect. This will make the drug development process and then the clinical trials process much safer and more efficient. So their policy wouldn't work because they're keeping the development of drugs to treat humans at a slow pace, and it can be a lot faster by using embryonic stem cells for research. And they're keeping the medical field just at a slow pace instead of making it a lot more efficient. And their policy also has disadvantages, like Lawrence Bonchek from Federal Funding on Stem Cell Research and the War Against Disease from CCSNet said of the 60 existing stem cell lines that this administration initially claimed were fully adequate for research, at most only 10 to 15 are usable. Worse, none can be used to develop treatments for humans, since all were originally grown on mouse cells, and they could expose humans to an animal virus that our immune system could not fight. We also know that adult stem cells, though readily available, are not as versatile as embryonic stem cells. So one of the disadvantages is that they said that they have 16 existing stem cell lines, but only 10 to 15 are usable. But not even those can be used because they were grown on mouse. So that's another disadvantage. And they also use induced pluripotent stem cells as an alternative. And Taisha Rowland of All Things Stem Cell says reports have caused some researchers to label iPSCs, which are induced pluripotent stem cells, as man-made cancer stem cells. The iPSCs indeed, by definition, can create teratoma tumors, which are tumors that can contain teeth, organs, and limbs. And when injected into animals and express many embryonic proteins, which allow them to differentiate into multiple cell types, just like tumors. So one of the disadvantages is that they can put the patient's health at risk by causing them tumors. And then another alternative that they use instead of embryonic stem cells is amniotic stem cells. Rick Ways from Scientific Potential in Amniotic Stem Cells says there's not going to be one shoe that fits all, said Robert Lanza, scientific director at Advanced Cell Technology in Worcester, Massachusetts. We're going to have to see which ones are most useful for which clinical conditions. George Daly, a Harvard stem cell researcher, echoed that sentiment. They are not a replacement for embryonic stem cells. So you can't just try and eliminate the embryonic stem cell research, because using these embryonic stem cells can really help make better drugs to treat humans, and that's why we should keep the existing policy.